have a discussion. Our next speaker will be Dr. Scott Ayton, who heads the Translational Neurodegeneration Laboratory and is the Deputy Director of the Melbourne Dementia Centre here at the Florey. Scott has a particular interest in ion neurochemistry and the regulated cell death pathway called ferroptosis, and today he'll be telling us about metal ions in cerebrovascular physiology and pathophysiology. Thanks.
Hello everyone. Thanks for sticking around for my talk, and thanks very much for the invitation to come here. It's been a really exciting day of work. I'm really glad to be a part of contributing to. Forgive my title, it's probably the most vague title I could come up with. It pretty much encompasses all of I guess cardiovascular physiology and pathophysiology, but I will be talking today about both aspects, about how metal ions can be involved in both the good side and the bad side of cardiovascular physiology. So I am a neuroscientist, but I keep on getting sucked into the vortex that is cardiovascular medicine, but I am not playing on my home ground when I am in this space. So I'm really thankful that I can come to this meeting where there are so many experts in the field. Also, I'm glad because when I go to talk about metal ions in neuroscience, I get a lot of people rolling their eyes and then going to sleep. But I feel, and maybe I'm wrong, but I feel that in cardiovascular there's a little bit more legitimacy with metal ions because, of course, metal ions like calcium and potassium and to a lesser extent sodium arbitrate the tone of cerebral blood vessels, and so there's no arguing that they've got a very important function there. And also on the flip side, when you've got hemorrhage, the iron in hemoglobin is a principal cause of toxicity. So metal ions really do have an important role for cardiovascular health and pathophysiology. So I'm going to talk about the good side and the bad side. We'll start off with the bad side. To begin on the bad side, I'm going to talk a little bit about an old story but new data relating to neurodegeneration. And so today, because we're amongst friends, I really want to talk about some of the most latest data that we've generated. Some of it's a little bit premature and early, but I hope I can get some feedback from you experts. This data was published this week actually, and it describes that iron is elevated in Alzheimer's disease. So here we have looked at a large postmortem cohort with collaborators in Rush University in something called the Memory and Aging Project. And we looked at iron levels in people who had died in this community-based cohort, and there was about 200 or so people in this cohort. When they entered the study, they did not have Alzheimer's disease, they didn't have cognitive impairment, and they signed up to annual cognitive assessments and they agreed to donate their brain when they died. And we categorised these people based on a clinical diagnosis of Alzheimer's disease and a pathological diagnosis of Alzheimer's disease. And while these overlap quite often, there's a significant number of people that do not have the clinical diagnosis of Alzheimer's disease despite having higher levels of pathology. And conversely, there are some people who are misdiagnosed as Alzheimer's disease. That means that they have cognitive impairment or dementia, but they do not have the symptoms of, they don't have the pathology of Alzheimer's disease. And you can see this here. So on the x-axis here is grading according to NIA-Reagan criteria on severity of Alzheimer's pathology, and we've also looked at several other scales which showed a very similar result. Also, we categorised them according to a clinical diagnosis of dementia. The purple are that they have dementia, and the white is they do not have dementia. And so you can see here that there are some people with low pathology, but they do have dementia, and some people with high pathology, and they do not have dementia. There's a lot of information we can take from this slide. Firstly, we can see that those people who have a clinical diagnosis of dementia and also have high pathology of Alzheimer's disease have elevation of iron. Yet those people who have got the high pathology of Alzheimer's disease yet do not have the dementia, do not have iron elevation. So it tells us that iron increase in Alzheimer's disease is not simply due to pathology deposition, but it actually better tracks with the clinical syndrome of the disease rather than the pathology deposition. While iron is increased, that's been reported many times before, but is this important? And so we are interested in this because iron is, it can be quite toxic. It can contribute to oxidative stress by the Fenton reaction and also participate in a cell death pathway called ferroptosis, which I will describe to you in a second. So iron is a way that you can have toxicity in Alzheimer's disease. We wanted to know, is this an important mediator of decline? So one way of looking at this, and I'm now summarising up a whole lot of data, is that we took the amount of iron in the brain and see if that correlated with the history of clinical deterioration. And here we've got a cohort of people. These people all had Alzheimer's pathology in their brain at the time of their death. And this is the year, this is annual assessments in the years prior to their death. So -12 to the time of which they died, which was 0. And this is change in cognition. And here, just for visual display, I've stratified the cohort into tertiles of low, medium, and high iron. And quite clearly, you can see those people with high iron had an accelerated cognitive deterioration upon their underlying pathology. If the patients did not have this pathology, there was an effect on iron, but it was very moderate compared to this effect. And this effect here was on top of correcting for other correlates of cognitive decline, including different types of vascular disease, Lewy bodies, tangles, plaques, ApoE4, and all the other clinical variables. And in fact, it was the strongest association we observed. We have also looked at the same, we've also found the same observation in other cohorts, and again, summing up some data where we used quantitative susceptibility mapping. It's an MRI technique that is sensitive to iron levels, and we looked at whether this could be prognostic for clinical deterioration in people who had been had amyloid identified by PET. What we can see here is we've stratified the cohort into those people who are amyloid positive and those people who are amyloid negative. And this here is the change in cognition over a 7-year period. So it's annual cognitive deterioration. And this is the hippocampal QSM. And what you can see is that those people who have underlying amyloid pathology and elevated QSM were associated with a more faster disease progression than those people who had low QSM. Conversely, if you did not have amyloid pathology in your brain, there wasn't an association between the QSM value and cognitive deterioration. So it's that combination of having underlying pathology, this is when iron becomes an important risk factor for deterioration. Finally, another approach we've looked at is by looking at ferritin in the CSF. Ferritin is the major iron-binding protein of the body, and ferritin in CSF is a reporter of the amount of iron within your brain. It also reports inflammation, and we've taken steps to correct for that. But here we've looked at change in FDG. FDG is a measure of brain activity and also brain atrophy. And we've stratified this cohort according to pathology by a CSF marker, which is the tau A-beta ratio. And those people who have high tau A-beta, so high pathology in their brain, and also high ferritin, were associated with a faster decline in FDG compared to those people with low ferritin. So what we see here is that those people with high iron in 3 different cohorts measured in 3 different ways have got a more faster deterioration compared to those with low iron. So what we see here is that the level of iron matters for predicting clinical deterioration in Alzheimer's disease. Is it cause or is it effect? Could iron just be elevating in those people who are declining faster, or is iron somehow contributing to the disease? We tend to think the latter because there are diseases of iron accumulation that result in disease of neurodegeneration. This is a class of disease called neurodegeneration with brain iron accumulation. So if iron accumulation occurs in Alzheimer's disease, it's quite reasonable that that will also contribute to neurodegeneration. But what I want to make the point at is that not everyone with Alzheimer's disease has high iron. But what we have observed here is that the amount of iron is related to how quickly you decline. And we take this clinical information and I guess think mechanistically about what could be going on within the brain, and we're thinking about what is the pathway of neurodegeneration in Alzheimer's disease. And we have the hypothesis, based on several lines of evidence, that the pathway of neurodegeneration is a cell death pathway called ferroptosis. This is a pathway that was just described by Peng Lei. I wanted to point out in this pathway that iron is one factor in this pathway. And there are multiple factors in the pathway. It's just named ferroptosis, but it could have been named lots of other components of this pathway. And what I wanted to point out is that ferroptosis is not how iron kills cells. We're not talking about a poisonous excess of iron and that just leads to cell death via this pathway. What we're talking about here is that ferroptosis is the way in which the cell recruits iron within itself so to commit suicide, to kill itself. So the amount of iron within a cell with low iron can still die by ferroptosis. You'll just need a higher signal. So iron is a moderating factor for susceptibility to ferroptosis, not dissimilar to what we found in our clinical results, where iron is also a moderating factor for clinical deterioration. So it agrees with the hypothesis that the mechanism of neurodegeneration in Alzheimer's disease is ferroptosis, because we can see iron as a moderating factor. We'll talk about this. So I guess we want to know what does cause iron elevation in Alzheimer's disease. There are several factors, and I've just listed a couple of them here. We can think that cerebrovascular changes could cause iron elevation in Alzheimer's disease. There are two main ones that I can think of. Hypoxia is well established that leads to iron accumulation via the HIF1 alpha pathway. You could also have hemorrhage that leads to iron accumulation because hemoglobin has got the most amount of iron within the body. Red blood cells have got just a huge amount of iron. This can cause inflammation, and inflammation also affects iron. I won't talk about that today. And you've also got metabolic and genetic that I'm not going to talk about. But of course, today we want to talk about cerebrovascular and how that might be leading as one possible explanation for iron changes in Alzheimer's disease. So one of these factors is because red blood cells, as I mentioned, has just got a huge amount of iron, and if there is a leakage, then it's very reasonable to assume that this is going to lead to local iron changes. One way we looked at this is to look at the Biofinder cohort. This is a cohort based in Sweden, and we worked with Oscar Hansen, and this is quite a large cohort of people with CSF taken, and you can see across these different diseases. When it says positive, that means positive on A beta. Excuse me. So what we looked at is many different factors associated with cerebral vascular pathology and inflammation and iron metabolism that might be able to explain iron changes and also prognostication for these different diseases. The caveat here is that vascular dementia, which is some of our most interesting results, is also our smallest group. So this work is, I guess, there's a caveat associated with that. So these are some early changes, early things that we've discovered. I'm not going to obviously go through with this. I just want to go through all this, but I just wanted to show you these are several different proteins we've looked at related to clotting and vascular pathology and also inflammation. And the strongest effect size we saw was for a protein called haptoglobin in vascular dementia. If we look at this a little bit closer, what we can see here is this is the odds ratio of CSF haptoglobin, and you can see that a standard deviation shift in haptoglobin caused an odds ratio, had a threefold shift in the odds ratio of having vascular dementia. Also, several other diseases, including Alzheimer's disease, also had elevated haptoglobin levels, but it was less than that of vascular dementia. So what is haptoglobin? Haptoglobin is a protein that binds hemoglobin. Hemoglobin is obviously comes from red blood cells, and it is a way that the body can remove the toxicity associated with the hemoglobin. So haptoglobin binds hemoglobin, and this is able to be taken up by the cell by the scavenger protein CD163. And this is a way we can remove hemoglobin levels. So haptoglobin is an acute phase protein. It elevates during the disease, during inflammation that can mop up hemoglobin. So we considered, could haptoglobin be a reporter of vascular damage? And so we stratified our cohort into haptoglobin positive and haptoglobin negative. And what we found is that everyone with vascular dementia were positive for haptoglobin. Which may agree with the concept that maybe haptoglobin could be reporting vascular damage. And then if we look at Alzheimer's disease, also these people were proportionally more likely to be high haptoglobin positive. And if you look at the healthy control people without any amyloid, there were some people who were haptoglobin positive, but this was less frequent compared to all other neurodegenerative diseases. So when I compare this to a large cohort that has been investigated post-mortem, we can see that some of the proportional changes that we see are similar to what is observed when this has been looked at post-mortem. This is a large cohort of people with Alzheimer's disease. At death, they observed vascular pathology in 80% of cases. Now, our cohort is much younger because it's not at post-mortem, and we see it less, maybe about 2/3 of people, but it may agree that we're seeing a vascular pathology. Also, cerebral vascular disease, everyone, of course, has got vascular pathology, and the unremarkable brain, they said 2/3, and we found about 1/3 of people had vascular pathology. So this is the early days for this concept, but we're thinking that maybe we can use this as a biomarker of vascular pathology, and I'd be very interested to hear your thoughts on that idea. Finally, I want to go really quickly into the good side. So we're looking at how metalines could also regulate vascular tone, and this is of course well established. Calcium is the arbitrator of contraction in a blood vessel, and potassium is a strong regulator of polarization as well as sodium. And what I'm going to suggest to you is that zinc, we've discovered, is also a regulator of vascular tone. So zinc is regulated by many different transporters. It's very difficult to genetically interfere with zinc, and you can't just add zinc on the outside because it's regulated by a very sophisticated mechanism. So one way you can do this is use a zinc ionophore, where we got a drug that binds zinc that is able to transport it into the cell. And what we found is that zinc ionophores are very potent vasodilators. Am I running out of time? Oh, no, sorry. So here is an example of a contracted smooth muscle cell. This is concentration of drug. We compare that this zinc ionophore has got comparable efficacy to acetylcholine and sodium nitroprusside, which are classic vasodilators. And so these zinc ionophores are very effective in causing vasodilation. Very unexpected result. And we can compare a bunch of different zinc ionophores. They all have the same effect in causing relaxation of blood vessel tone. Zinc alone has no effect because you can't really shift zinc just by adding it alone. So you wouldn't expect the zinc levels in your plasma, for example, to affect your blood vessel contraction. So you need to have a drug that delivers that. We've confirmed this result in human blood vessels, where we see that zinc ionophores also are very effective in causing vasodilation, and also in a rat, where we can administer this drug and cause lowering of blood pressure. But this is where it gets kind of interesting and exciting, is that when we use the reverse effect, when we use a zinc chelator, we can cause contraction in certain blood vessels, particularly cerebral blood vessels. So if we use a zinc chelator, we can get full contraction of cerebral blood vessels, and this is not the case in non-cerebral blood vessels. So it may be another way of targeting cerebral blood vessels by targeting zinc biology. But it also tells you that zinc is having a role in causing the resting state relaxation of blood vessels. And so this demonstrates a biological role for zinc in the maintenance of blood vessel tone. So there's a lot more to say about that. That's a really huge project, and we've discussed the mechanism. We're almost there demonstrating the mechanisms. It's via two mechanisms. One is via the sensory nerve, where zinc, I'll have to talk about this another day, and also there's a mechanism directly in the smooth muscle. So zinc, we're showing, has got a very important role in cardiovascular physiology. This work has been done by Ashinafi. He's the smallest figure in this acknowledgement slide, but he's done a huge amount of work with that, and he's sitting in the audience. And also several other collaborators within the Florey and the University of Melbourne. And our work in the clinical cohort stuff was done by people at the Florey, particularly Ashley Bush and others. And our collaboration with Rush University, ADNI, ABLE, and CSIRO. Thank you for your time.
Fantastic, really exciting work, Scott. I'm looking forward to some discussion at the end. I want to know more about haptoglobin, that's for sure.